Hi and Assalamualaikum. My name is FAIZAH binti ZAINAL ABIDIN and my matrix number is 208074. I'm going to do the hands-on assessment for BMY3201 and I'm going to demonstrate step by step the technique for gram staining. The two major groups of bacteria can be divided into gram-positive and gram-negative. The gram stain technique is based on differential structure of the cellular membranes and cell walls of the two groups. Gram-positive organisms contain highly cross-linked layer of peptidoglycan that retains the primary dye, crystal violet, following the application of the modern iodine. The iodine and crystal violet form a complex within the peptidoglycan. When decolorizer is applied to the cell, the CVI complex remains within the cell, making it appear dark purple to blue. The gram-negative organisms do not contain a thick cross-linked layer of peptidoglycan. The peptidoglycan is loosely distributed between the inner cell and the outer membrane. Following the application of the crystal violet and iodine, the CVI complex are not trapped within the peptidoglycan. Application of the acid alcohol decolorizer dehydrates the outer cellular membrane, leaving holes in the membrane and effectively washing or removing the CVI complex from the cells. Cells appear colorless. To make the colorless cell visible, a secondary stain, safranin, is applied, leaving the gram-negative cells big. Four reagents used in the gram stain is primary stain, mordant, decolorizing agent, and counter stain. The primary stain we have crystal violet. Its function is to impart the color to all cells. For the mordant, we use the iodine. Its function is to intensify the color of the primary stain. The 95% ethyl alcohol is used as decolorizing agent to establish a color contrast. The last reagent is the counter stain, which we will use the safranin. Its function is to contrast the color of the primary stain. In this experiment, you will need culture, a slide, inoculating loop, the reagent, the alcohol, and the tap water. The procedure will carry out by doing a smear preparation first. Before we prepare a smear, we have to do the aseptic technique by flaming the loop into the flame and cool it. The smear was prepared by placing a drop of water on the slide. By using a aseptic technique, the organism from the culture was transferred to the drop of water. A minimal amount of culture is required. If culture can be detected visually on inoculation loop, it indicates the collection of too much culture. The organism and water were mixed, then it was spread by means of a circular motion of the inoculating loop. The smear was allowed to air dry and then heat fixed by passing the smear over the flame several times. Heat helps the cell adhesion to the glass slide and prevent the significant loss of culture during staining. Firstly, the smear was flooded gently with crystal violet for 1 minute. Then, gently wash off the stain with tap water. After that, gently apply grams iodine for 1 minute. This step is known as fixing the dye. Wash gently the iodine with tap water. Then, the smear was decolorized with 95% ethyl alcohol by adding the reagent drop by drop until the alcohol runs almost clear, showing only the blue tinge. This step is known as solvent treatment. And again, rinse with the tap water. The smear is counter-stained with safranin solution for 45 seconds. And again, rinse with the tap water. Excess water is blotted with bibulous paper. Finally, examine the culture under oil immersion. Expected result: the gram-positive bacteria will appear blue or purple in color, as it has the ability to retain the crystal violet dye during decolorization. While for the gram-negative bacteria, it will appear pink or red, as it loses the color of the primary stain during decolorization, and the safranin that were used as a counter stain will give the pink color to the decolorized gram-negative bacteria. For easier identification,